Hello to all our attendees. We're just waiting a little bit, a couple of minutes till it's the right time. So we will start briefly. So now it's time. Hello and welcome everyone to another webinar here in Mercodia. My name is Miguel Guillermo Sainz, and I'm a product manager and the medical science liaison at Mercodia here in Uppsala, Sweden. And I will be your moderator today. I am very glad to see that actually we have quite a lot of attendees at the webinar with Dr. Megan Capozzi and Dr. Kimberley L. And they will present some of their work today. Both of them are postdoctoral fellows and researchers at Duke University at the Duke Molecular Physiology Institute. Megan received her PhD from the Vanderbilt University in 2016, and Kimberley received her PhD from Indiana University School of Medicine in 2018. Both of them, they work in the laboratories of Dr. Campbell and Dr. D'Alessio. Now, during the webinar, all attendees will be able to write questions to Dr. Capozzi and Dr. L by typing your questions in the panel that you see on your screen, and your questions will only be visible to us as the organizers and not for other attendees. After the talk, if times allow, we will try to read through all of the questions. And if there are questions that we have missed, we will send them to the presenters and we will send you back the answers later on. Now, the title of this webinar is "The Alpha Cell is Critical for the Full Incretin Effect." And it is a pleasure for me and for all the Mercodia team to give the webinar platform to you, Megan and Kimberley. So Megan, please, the floor is yours.
Thank you. Thanks for that introduction. And yeah, I'm excited to talk to you about our recent work and kind of how it's all come about. So a brief outline of what we're going to talk about today. We're just going to give an overview of what alpha cell to beta cell communication is. And then I'm going to talk to you a little bit about the more pharmacologic ways that we've kind of played around with this system to look at postprandial glycemia. And then I'm going to pass it over to Kim, who's going to show you her recent and really interesting work and how it pertains to normal physiology and particularly GIP. So just to start us off on something we know, the canonical view of glucagon signaling. So glucagon is considered a hypoglycemic responsive hormone that is secreted from the pancreas and the alpha cell in the pancreas whenever there's conditions of hypoglycemia. It can then go talk to the liver where it stimulates either glycogenolysis or gluconeogenesis in order to elevate glycemia. And as we know, insulin is kind of the opposite here. So insulin is secreted from the beta cell of the pancreas, where it's secreted in more hyperglycemic conditions where it talks to the liver and a number of other target tissues to stimulate uptake of glucose. And so that's really led to this opposing view where glucagon and insulin really shouldn't be elevated at the same time. And that's even been pushed further with the concept of the glucose tolerance test. So this is one in humans, an older test here where they gave a carbohydrate-rich meal. And this is our, again, canonical signaling, where when we give a large carbohydrate meal, we see glycemia elevate. And that comes with a steep rise in insulin to bring glycemia back down. And glucagon decreases. And so that's kind of what we're used to when we think about meal tolerance, but that really only focuses on one component of the meal. And so if we actually think about what we normally eat, we usually don't eat just straight carbohydrate. Normally, we're also incorporating other nutrients in our meal. And so in this study, they also include another nutrient, protein, which is known to stimulate the alpha cell. And it's pretty interesting when they look at these same parameters, you see glycemia doesn't really move when you give a protein-rich meal. You do see an increase in insulin, although not to the same level as in response to carbohydrate. But you also see this really large rise in glucagon as well. And so glucagon and insulin are both elevated at the same time. And we think that this is probably, normally you're eating carbohydrates, proteins, a whole bunch of different nutrients. And we think it's probably likely that insulin and glucagon are both being secreted in response to normal feeding. And so we started looking at how glucagon and insulin might be interacting at the beta cell. And we did this because glucagon is secreted from the alpha cell, which is right next door to the beta cell. And the glucagon receptor is expressed on the beta cell as well. And what's interesting with the glucagon receptor is that it acts much like the GLP-1 receptor in that it's a GS-coupled receptor that stimulates cyclic AMP. And this is pretty interesting when we think of GLP-1 because we think of GLP-1 as a glucose-lowering agent and an insulin secretagogue. And so that opens up the idea that glucagon might be doing the same thing. And so when I say these are glucose dependent, we usually think of this system as like kind of a dimmer switch. So you need glucose to turn on the beta cell. So you need a beta cell on. But then once you include a ligand to start elevating cyclic AMP levels, it acts almost like a dimmer. So you can kind of turn up the insulin response. And so just to show you that glucagon works like GLP-1 and that it only stimulates insulin at high glucose concentrations, I have this experiment here. I'll spend a little more time talking about these graphs because you'll look at a lot of these between Kim and I today. But if you focus on insulin here on the y-axis and the various treatments here on top of the graph, when we stimulate with glucagon at a low glucose concentration, you have no insulin secretion. But then once you turn that beta cell on with glucose, shown here, when we give that same dose of glucagon, now you see a much larger rise in insulin secretion. And so we sought out to just ask the question, how important is glucagon for beta cell function? And so, like I said, I'd take a minute to introduce you to the islet perifusion technique because we're going to spend a lot of time looking at these graphs. So to do this, we generally, I think most of the graphs are using mouse islets. We can do this with human islets as well. We isolate islets from the mouse, put them into these chambers that then we put into a biorep perifusion machine. So each chamber is an N of 1. And then we have buffers that go in here, and we can control the flow rate and which buffers go over our islets for how much time. It's then the buffers will flow over the chambers and then drip into 96 well plates. So we're collecting hormone levels on a minute-by-minute basis using this machine. And so that's what the data will look like here. Generally, so each chamber is an N of 1, like I said, and generally our graphs are from about an N of 6. We look at different hormone secretion that's going to be shown on the y-axis, and then we're following our data on a minute-by-minute time points on the x-axis. And then you'll see whatever we're treating with is shown on top. Excuse me. So here we looked at glucagon-stimulated insulin secretion, and like I said, glucagon stimulates insulin secretion at high glucose. So when we go from low to high glucose in wild-type islets, and then go to increasing levels of glucagon, we see increasing levels of insulin secretion. And so we wanted to see what the contribution of glucagon through the glucagon receptor was for insulin secretion in this paradigm. So we generated a glucagon receptor knockout specifically from the beta cell to ask these questions. And when we put that mouse or those mouse islets through this perifusion, we were pretty surprised to see that there is no impairment of insulin secretion in response to glucagon. But there was evidence in the literature that glucagon can actually bind the GLP-1 receptor as well. And so we tested this using a drug to block GLP-1 receptor. It's called exendin 9. And that's shown here in green, where whenever we now increase glucagon concentrations in the presence of a GLP-1 receptor antagonist, we largely blunt glucagon-stimulated insulin secretion. And then we thought that maybe this residual insulin secretion that you see as we go in increasing doses might be the residual contribution of the glucagon receptor. And so when we give exendin 9 to the glucagon receptor knockout, we can pretty much completely blunt glucagon-stimulated insulin secretion. And we've done this a couple of different ways with drugs with the inverse knockout. So this is a pretty consistent finding that glucagon works through both receptors, but predominantly the GLP-1 receptor. So you'll notice the doses I'm using for glucagon are pretty high here, 300 nanomolars, a high amount. So we wanted to use a paradigm where we're making the alpha cell secrete physiologic levels of glucagon. And I'm not showing you all the back work for this, but we use different amino acids to do that. So glutamine and arginine at the doses used here in wild types will stimulate the alpha cell to secrete glucagon that can then talk to the beta cell. And so what I'm showing you here in just wild type islets is we go from low to high glucose and then use these amino acids. And then we wanted to test this now instead of blocking the receptors, getting rid of the ligand. So we used proglucagon knockout mice. So they don't have any proglucagon products coming out of the alpha cells. So no glucagon, no GLP-1. And when we do this, now we can completely block amino acid stimulated insulin secretion, which we had hypothesized, but what was really surprising was that we actually block glucose stimulated insulin secretion. So we didn't necessarily expect that. We thought that would be independent of the system. And this finding might look like, maybe you just have really messed up islets because of the lack of proglucagon products. But when we add back in different ligands, so glucagon, GIP, GLP-1, to bolster the cyclic AMP levels, we can get completely normal insulin secretion. Also, to show you that this isn't just a phenomenon we see in mouse islets, we can do this in human islets as well. So blocking here the receptors using exendin 9 and a glucagon receptor specific agonist. In the humans, this system may be even more important because we can block insulin at low glucose as well as increasing glucose concentrations. So this does seem to be important, not just in a mouse. So all of the data I've shown is now in isolated islets where the ligand's only coming from the alpha cell, but we can put these into, or we wanted to now take this work into whole body glucose metabolism. And again, I'm going to play with like pharmacology more here. So here we're giving a really large dose of glucagon, and this is kind of that canonical view of glucagon. When you give glucagon to an overnight fasted mouse, their blood glucose is pretty low at 75 mg per deciliter. We give glucagon, we see a nice rise in glycemia. But knowing now that this can kind of work like an incretin almost in a fed state, we tried the same large dose in a fed mouse, and we're actually able to lower glycemia when we give the same dose of glucagon. And that's largely because of insulin. So where you don't have your beta cell on in a fasted state, you really get minimal increases in insulin. But you can see when you have a beta cell on situation in a fed state, now you could get about a 4-fold increase in insulin secretion. Kind of another cool trick we did with this was to try to dissociate now glucose from the equation, but just kind of focusing on this beta cell on state. And so to do that, we used a sulfonylurea called tolbutamide. So instead of having glycemia elevated, we used tolbutamide to open the KATP channel and have the beta cell on. But since the beta cell's on and secreting insulin, we actually have lower glycemia. So we're starting actually at around a fasted level. But now, since we turned the beta cell on, when we give this large dose of glucagon, we actually lower glycemia. And again, that's happening with a really large rise in insulin secretion. And so this is really important when we think about treating patients. So this is an example from a case study with a 50-year-old man who had no history of hypoglycemia, but had taken a handful of sulfonylureas. So it gets really hypoglycemic, as you'd expect. Continuous glucose administration did not restore euglycemia, so the doctors gave a big dose of glucagon as a rescue agent. And based on the data I've just shown you, it's not too surprising that while glycemia is really temporarily elevated, it also went right back down, and you had, again, severe hypoglycemia that was probably largely caused from this massive rise in insulin that we would predict from our model. So it's really important to understand how these hormones are communicating for patients. To show you now that this is, and put this in the context of alpha to beta cell communication, we generated a mouse that lacks alpha to beta cell communication by deleting the glucagon receptor and the GLP-1 receptor specifically from the beta cell. So now the alpha cell can't really talk to the beta cell. And we used this 1 mg per kg dose model with lowered glycemia in wild type mice. But whenever we do this in our knockout mice that don't have alpha to beta cell communication, you no longer mount an appropriate insulin response, and that leads to this elevation in glycemia. And to take it just one step below closer to normal physiology, we put this mouse through a glucose tolerance test. We had to push them with a high-fat diet to have some metabolic abnormality. But whenever we do this now and give an IPGTT or OGTT, you see that glycemia is significantly impaired in these mice. So alpha to beta cell communication is pretty important just for normal glucose tolerance. And so with all that kind of pharmacology behind us, I'm going to switch it over to Kim, who can tell you more about GIP and this whole system in normal physiology.
Can you see that? Is that working?
Yes.
It doesn't. Okay. So thanks, Megan. So now that she's convinced you that glucagon is important for insulin secretion, I'll be bringing you the sequel or the prequel, which is when and how is that glucagon secreted. And she left off kind of explaining this in the context of a technique that we use called glucose tolerance testing. We give various nutrients via various routes of administration. And here we're interested in the glucagon response to these various fed states. So this is a nice experiment that Megan did here. Let me see that. So she gave an oral gavage of different nutrients. So she started with PBS as a control, and we see that insulin and glucagon don't change. When she gives oral glucose, we see a spike in insulin secretion after 10 minutes, but again, glucagon doesn't really change. And then she gave a mix of nutrients, so glucose as well as others, and she saw that a rise in insulin and glucagon after 10 minutes. And if we compare the correlated blood glucose curves between these different nutrients, we can see that mice given just glucose, although they have a huge spike in insulin, they actually have a higher blood glucose curve, and so they're a little more glucose intolerant compared to mice that get a mixed meal that are able to lower their blood glucose early on in this challenge because they have insulin and glucagon. And furthermore, now this is in human, but compared to an oral glucose, when you give mixed meal, you can actually get much more GIP and GLP-1 in a mixed meal. So the only difference here is that the kinds of nutrients that we're giving, a mixed meal not only has glucose, but it has protein and fat. And this more closely resembles a meal that we would normally eat. So although sometimes we do like to eat a handful of candy, a mixed meal that has glucose, protein, and fat more closely resembles what we normally eat. So now that we've established that we can stimulate glucagon by feeding a mix of nutrients, we can really dial in and focus on what actually stimulates the alpha cell to secrete glucagon. And so for the past 50 years or longer, it's been known that amino acids stimulate glucagon. So when we eat protein, amino acids are absorbed in the gut here, and they make their way to the pancreas and they stimulate glucagon. These amino acids are alanine, arginine, glutamine, and glycine are the main glucagonotropic amino acids. And these amino acids stimulate insulin as well, but only at high glucose. And branched-chain amino acids actually do not stimulate glucagon. When we eat, we also stimulate the secretion of GIP from the gut and GLP-1, but GLP-1 actually inhibits glucagon, whereas GIP will stimulate glucagon. This glucagon that's stimulated by GIP is actually a pretty small response, and it depends on glucose levels, so it doesn't happen at high glucose. So here are some examples of GIP stimulated glucagon secretion, and what I'll just point out is that here there's not a whole lot of range. So the range of increase is only about 4 to 5 picomole in humans and in rats, and so it's not a huge effect. And so our hypothesis was informed by what we know about the beta cell. So if we give glucose to an islet, we get insulin secretion. That's classic glucose-stimulated insulin secretion, GSIS. When we give GIP as well, we get potentiated insulin secretion. And so that potentiated insulin secretion can only occur when glucose is present. So we thought of the beta cell as needing a similar kind of condition. So it needs something to trigger the alpha cell. So right now it's inactive. So we hypothesized that instead of glucose, in fact, amino acids are the major secretagogue for the alpha cell, and that when amino acids are present, GIP can potentiate glucagon secretion, which, as Megan showed you, is important for insulin secretion. So now I will show you again more perifusions. So at low glucose in a perifused islet, glucagon is pretty steady, and that's our baseline secretion that we compare it to. So when we give islets alanine, we see a rise in glucagon secretion. It actually resembles the shape of GSIS. So you get a first phase and a second phase secretion. Now, if we focus on this area here where the cells have found an equilibrium, we actually see we get only a modest rise in glucagon secretion. Now, when we give GIP, and this is all low glucose, we again get this rise in glucagon, and I've zoomed in here, but it's only about 2 to 3 fold increase from the baseline. Now, as you might guess, when we combine the two, so we combine GIP and alanine, so the hypothesis is that we're triggering the alpha cell to be able to secrete glucagon, and that GIP could potentiate that. What we see is that when we do that, we actually do get the synergy between amino acids and GIP. And so, and it's what we call synergy in that we get more secretion than the individual treatments by themselves. So 9 plus 7 is not 82. So we get a lot of glucagon secretion. So to confirm the mechanism of this synergy, we generated an alpha cell GIPR knockout. So we took out the GIPR and the alpha cell specifically. We went back to our perifusion protocol, and at low glucose, we give GIP or GIP plus alanine, and we see our synergy. But when we remove the alpha cell, we don't see that GIP can potentiate the alanine-stimulated glucagon. So we lose that synergy for glucagon. So we can also show this in vivo. So when we inject mice with GIP, we get a modest rise in glucagon. Again, inject them with alanine, a slight increase. When we inject them together, we get more glucagon secretion. And to confirm it again in our knockout mice in vivo, we've taken out the GIP receptor, and we see that we lose that synergy in the alpha cell knockout mice in red. And so the glucagon secretion that we observe is kind of analogous to what we see in the beta cell. So in the beta cell, GIP will potentiate glucose-stimulated insulin secretion. And if we think of the alpha cells in a similar capacity, then they also need a fuel to activate or to trigger. And so we think that fuel is amino acids. And so in an alpha cell, GIP potentiates amino acid-stimulated glucagon secretion. Now, everything I've shown you is at low glucose, which is where the alpha cell is kind of happy. But what does this really mean for insulin secretion? Because insulin secretion requires high glucose. But classically, glucagon secretion is dampened by high glucose. And we've seen this over and over. We give high glucose, glucagon drops. And actually, at this level of glucose, alanine stimulated glucagon is also blunted. And so if insulin secretion requires high glucose, what does this really even matter if glucagon is dampened? What does that synergy mean? And so does GIP and alanine have a synergistic effect at high glucose? And if so, does this translate to synergy for insulin secretion? And so again, I applied our perifusion protocol to wild type islets, and I did all of this at high glucose now. And so given high glucose, we see that the baseline glucagon secretion drops. We treat with alanine, we see a slight blip, but it doesn't really work at high glucose. GIP can stimulate it a little bit at high glucose, but not a lot. But when we give them together, we do see that we still get synergy for glucagon secretion. So that's glucagon, but how do these affect insulin? So we would think that when we give amino acids, that triggers the alpha cell to secrete glucagon, and with the treatment of GIP, we would also get potentiated glucagon. And as Megan explained, that's important for insulin secretion through actions on the GLP-1 receptor and glucagon receptor in the beta cell. And so indeed, when we measured insulin secretion in these samples, we saw that GIP and alanine together can increase insulin secretion beyond what we would get by these treatments alone. So it would seem that as long as the alpha cell is activated or triggered, GIP and alanine can induce alpha to beta cell communication at high glucose. Now, we wanted to confirm that this was alpha to beta cell communication, and so we tested this mechanism by blocking the GLP-1 receptor. So Megan used this as well. She used extendin 9 to block the alpha to beta cell communication. And so we interrupt the ability of the alpha cell products to talk to the beta cell. So here in our wild type perifused islets. We give GIP in both control or exendin-9 treated groups. And because GIP works through the GIP receptor on the beta cell, we didn't expect a change in this condition. But when we give the control islets here on top, we give alanine, we see our synergy in the presence of the GLP-1. So we've got the alpha to beta cell communication present. But when we do treat with exendin-9 and we block the GLP-1 receptor, we're blocking the ability of any glucagon to be able to talk to the beta cell. So what we see is that we lose that alpha to beta cell communication piece and we don't get insulin secretion. So now I want to kind of pivot and show you what happens when we take this to a whole body situation. So we subjected high-fat fed mice to a IP glucose tolerance. So we've injected glucose. We don't rope in the gut peptides, so we just have glucose. In this setting, the nutrients bypass the gut, and we would hypothesize that without amino acids, the alpha cell is inactive, it can't be triggered. Any glucose disposal that we see during this test is mediated purely by GSIS in the beta cell. And so we would expect that a loss of GIP receptor would actually have really no effect. It's irrelevant. And so that's exactly what we saw. We saw that in response to an IP glucose tolerance, the knockout mice were not different from the controls. And so then we did an oral glucose tolerance test where we gavaged the mice. So the nutrients pass through the gut and are able to stimulate the gut peptides. So we've got glucose and GIP in the mix. So this is what a control oral glucose tolerance test would look like compared to an IP glucose. So they're able to, excuse me, lower their blood glucose much more than an IP glucose. And that's the classic incretin effect. You get more insulin secretion because you have this GIP. Now, if we remove the alpha cell GIPR, again, the alpha cell has no amino acid input, so we would hypothesize that it is inactive and dormant. And so what we see is that these alpha cell knockouts, as we'd expect, don't have any difference in oral glucose tolerance test either. So now we go back to our mixed meal, which includes, again, glucose, fat, and amino acids. And we give that via oral gavage. So we've got all of those nutrients and GIP. And so the control mice actually became hyperglycemic. They became hyperglucagonemic and hyperinsulinemic, which is typically what we see in diet-induced obese mice. And when we take out the GIP receptor, we would expect, since the alpha cell is triggered in this situation, we would expect to see some kind of difference. So what we saw was that these mice actually became more glucose intolerant compared to the controls. They lose this glucagon response, and as a result, they lose any insulin response or a dampened insulin response. And so this all highlights how important the alpha cell is for glucose homeostasis. If you think about it, I've shown you that we lose insulin secretion when we knock out an alpha cell receptor. So that's pretty cool. So just to conclude and wrap up here, first I've shown you that GIP potentiates amino acid stimulated glucagon secretion. This happens at both low and high glucose. The synergistic effect that we see with glucagon is at high glucose, and this corresponds to an increase in insulin secretion. And finally, the loss of the alpha cell GIPR in combination with high fat feeding results in glucose intolerance and actually impairs glucagon and insulin secretion in response to a mixed meal. So yeah, that's our story. We're sticking to it, and thanks for tuning in. I'd like to thank the members of our lab, which is Megan and me, and then Sarah, Kanchi, Jen, Bree, and Ivan, and then our leaders here who've secured all of this funding for us to do all these experiments. So thank you. And Megan and I will happy to answer any of your questions now.
Great. So thank you so much, Megan and Kimberly, for great talks. And yeah, now we have a couple of questions here that they have been sent during the webinar, and I will start reading them to you. So hopefully, hopefully you will have nice answers. I'm pretty sure you will. So one of the questions that we have is, what is the relative importance of the GIP receptor on alpha and beta cells when considering the role of GIP in glucose homeostasis?
Kim, you want to answer that one?
I don't know who wants to answer that one.
Could you repeat the question? I'm so sorry.
Sorry?
Could you repeat the question?
Absolutely, yes. So what is the relative importance of the GIP receptor on alpha and beta cells when considering the role of GIP in glucose homeostasis?
Right, so GIP is an incretin, so when you give it, you actually get more insulin secretion, right? So the alpha and beta cells both have the GIP receptor. But I think like I showed in knockout studies, when we knock out GIPR in the alpha cell, we can still get we get insulin secretion in response to glucose, but not into a mixed meal. So all of this together just kind of shows the importance of both receptors on both cell types to mediate meal tolerance. So it's not just glucose tolerance.
I think to go along with that too is when you're thinking of the beta cell, there's a lot of other inputs for different incretin receptors. So you still have GLP-1, you have glucagon. Certainly you lose the GIP. If you lose GIP input, that's pretty important. But as far as we know, in terms of incretin receptors, at least the GIP receptor is the only one in the alpha cell. So it relies pretty heavily there, whereas you might have some compensation in the beta cell.
We have another question here that is, what are the implications for tirzepatide or other GIP agonists?
You can take that.
So tirzepatide is a dual agonist, so it acts on the GLP-1 receptor and the GIP receptor, and it's favored toward the GIP receptor, and so we can look at that kind of how it affects glucagon secretion, because tirzepatide can lower blood glucose, it lowers body weight, it's pretty good in vivo, but we don't know exactly how it works, so it could actually be just a super GLP-1 receptor. We don't know if it's working through glucagon, but it is a good tool that we can use. Yeah.
And we have a couple more. Sorry. So there is this question that says, so you mentioned that the glucagon synergy occurs in vivo and requires alpha cell GIP receptor. So is there a scenario where the GIP receptor could be inhibited or blocked in a physiological way, and that would result in a secondary effect?
Yeah, so we've used GIP receptor antibodies to block the GIP receptor in vivo. I don't know what that means for human studies, and I don't know how to target it specifically for the alpha. In low glucose situations where the alpha cell is more activated, and in that situation we could use and block GIP receptor.
And I think in terms of physiology at this point, I don't think we know, we know that GIP doesn't work, or the GIP receptor in beta cells doesn't work as well in diabetic patients. I don't think we're at a point where we know that for sure in alpha cells. or like what that means, because we also think that there might be some loss of alpha to beta cell communication in diabetic patients. And so how GIP plays into that is still kind of an open question.
Okay. And sorry, I'm just I was just thinking also right now that you were mentioning that. So, for example, what would be, or what could you think that could be an implication, for example, on patients that they are undergoing a bariatric surgery? If I guess that would be a completely extra thing to add to the discussion, though, right?
Yeah, well, I mean, so I know bariatric surgery patients definitely have a lot more glucagon, and I think, you know, a lot of times you think, oh, that should be a bad thing, but potentially, depending on, you know, glycemia and control of glycemia, control of different nutrients in the body, that potentially that hyperglucagonemia that you sometimes observe in bariatric surgery patients, that might actually be stimulating more insulin, or might have a different effect, I think, than what we normally think canonically for controlling glycemia.
Okay. And then there's another question here that says, can you discuss the role of GLP-1? Can you discuss the role GLP-1 has on alpha cells? From my understanding, the GLP-1 is inhibitory to glucagon secretion, but you're showing that GIP has the opposite effect. Is this effect you are showing of increased glucagon signaling only an effect during pharmacological dosing of GIP? How does your data relate to physiology?
So the alpha cell, it's a point of contention whether the alpha cell has GLP-1 receptors. And so the GLP-1 inhibitory effect on the alpha cell is mediated through the beta cell, I believe. So the insulin secretion would inhibit glucagon. Right. Is that right?
Yeah, and I think at least from our limited studies with the, we haven't done a ton with this, but at least in the perifusion system where we can kind of control, yeah, I don't think GLP-1 drastically reduces glucagon secretion in that isolated system. So it might be working in indirect mechanisms to do that. But I think, like with Kim's studies with using the alpha cell GIP receptor knockout, I know that, you know, that's not exactly, you know, completely normal physiology, obviously, with a knockout, but she did use just meal tolerance tests to manipulate this without giving pharmacologic doses of any incretins. That was just the normal secretion that you see in response to a meal. And so I think that does highlight that these do play a role in normal prandial conditions without having to inject like pharmacologic doses of everything.
Okay. Yeah, I mean, yeah, it's a lot of, I guess that there are a lot of different conditions that each one can put for this for the experimental setting, so to say, right?
For sure.
And there was another question that popped up that was, what would be the variability, especially thinking about animal islets, thinking about the age of the mouse, the strain, and the gender of the mouse? I don't know if you have any sort of.
Yeah, I mean, we've played with a lot of different islets. I don't think we've done a ton of different like strain comparisons, so I can't speak to that, but we've done comparisons of female and male mouse islets, and they are indistinguishable. And age, we really don't notice any difference with that. We haven't done like specific controlled experiments to look at that, but once the islets are in a dish, they look to be pretty comparable, which obviously is different than the in vivo situation. But yeah, in our islet perifusion, at least with C57s, males and females, age doesn't play a big factor.
Okay, that's good. I don't know if there are any other questions now. More questions from the audience? And if not, then I will say like, okay, if there are some other questions that they might pop up later on, then we will send them to you. And with this, I guess that we will finish the webinar for today. Thank you so much to both of you for taking the time to give this fantastic presentation and for answering all like the questions that popped up. And well, we'll be completely eager to know what happens in the future with your studies. And thank you again. Thanks to you, and thanks to all the attendees.
Yeah, thank you. Yeah, thanks for giving us a chance.
No problem. Bye.
Bye.